At the heart of modern biology is this amazing idea that DNA, RNA, and proteins are basically books written in an ancient language. But how do we actually read them? How do we find the shared stories and evolutionary secrets hidden inside? Well, today we're going to dive into one of the most fundamental tools we have for doing just that: pairwise sequence alignment.
So here's our game plan. We'll start with the big why. Why do we even bother comparing sequences? Then we'll clear up a super common point of confusion, the difference between similarity and homology. After that, we'll get into the how we measure the difference, explore the two main strategies for alignment, and finally, how to make sense of the score so you know if you've struck gold. All right, let's kick things off with the most basic question. Why do we do this? Why go through all this trouble? It really all comes down to answering one single, really powerful question. That's it. Are these two strings of biological letters actually connected somehow? Now, it sounds simple, but looks can be really deceiving. Just think about the words pair and tier. They look almost identical, just one letter off, but they mean completely different things. So in biology, we need a solid, rigorous way to figure out if similarity is just a coincidence or if it points to a real shared history. And that's exactly where pairwise sequence alignment comes in. It's our systematic way of lining up two sequences to find the absolute best possible match. And why does that matter? Because when we find those similar regions, we can start making some really cool educated guesses. Like maybe they do the same job, or have a similar 3D shape, or, and this is the big one, maybe they share a common ancestor. Okay, this brings us to a really, really important point. And honestly, it's one of the biggest mistakes people make in this field. We've got to be super careful with our words here because in evolution, they have very specific meanings. Listen up, because this is crucial. Similarity is just what you observe. It's a number, a percentage. It's you saying, hey, these two things look alike. Homology, on the other hand, is a conclusion you draw. It's an inference you make about a shared evolutionary past because you saw a significant amount of similarity. So, you measure similarity, but you infer homology. They are not the same thing. And homology, well, that's the real prize we're hunting for. When we say two genes are homologous, we're making a huge statement. We're saying that way back in the history of life, there was one single ancestral sequence, and both of the sequences we have today came from that original one. It's all about shared family history. And this family story has a few different flavors. You've got orthologs, which tell a story about new species forming. Think of the same gene in a human and a mouse. They split off when our species diverged, but they often keep doing the exact same job. Then you have paralogs. They tell a different story, one about gene duplication inside one organism. A gene gets copied, and now that new copy is free to evolve and maybe even learn a whole new trick, which is a huge driver of innovation for a species. Okay, so if we're trying to find similarity to prove homology, how do we actually, measure it? We need a ruler, right? A way to put a number on it. And in bioinformatics, we've got a couple of go-to rulers for measuring the distance between two sequences. Our two main rulers are Hamming distance and an edit distance. Hamming is the simple one. It just counts up the mismatches. But it has a huge catch. It only works if the sequences are the exact same length. But come on, evolution is messy. Genes get chunks added, bits get deleted, it's not that clean. And that's why edit distance is the real workhorse here. It's designed to handle not just swaps, but also insertions and deletions, the whole shebang, making it perfect for the messy reality of biological sequences. So, we've got our ruler, now we need a strategy. The question is, are we trying to compare two entire books from cover to cover? Or are we just trying to find one paragraph that they both have in common somewhere inside? This choice leads to two totally different ways of doing things. And those two strategies are called global and local alignment. Global alignment, as the name suggests, tries to match the two sequences across their entire length, from the first letter to the very last. It's what you use when you think two things are already pretty similar. Local alignment is more like a search engine. It doesn't care about the whole sequence. It just hunts for the single best patch of similarity it can find, no matter where it's buried. So you'd pull out a global alignment when you're comparing, say, a human gene to its direct counterpart, its ortholog, in a chimpanzee. You expect them to be almost identical from start to finish, and your goal is to find every little difference, every single mutation. This is how we track recent evolution or hunt down genetic markers for diseases. But what if you're comparing two proteins from, a person and a plant? They're going to be wildly different. A global alignment would just be a mess. But a local alignment? That could be amazing. It could find one small, critical section, a functional domain that's been saved by evolution for millions of years, even while the rest of the protein changed completely. Finding that shared piece is like finding a fundamental building block of life itself. All right, we're on to the final and maybe most important step. The computer spits out an alignment, but is it any good? How do we know if it's a meaningful discovery or just random noise? We have to turn that alignment into a number, a score that tells us objectively how good it is. So just look at these two ways of lining up the same sequences. I mean, which one looks better to you? It's tough to say, right? They both had to insert some gaps to make it work. We need a system, some rules to tell us which one is the winner. And that system is a scoring scheme. It's basically a simple game of points. We give points for every match, but we take away points for a mismatch or for having to create a gap. The alignment with the highest score at the end wins. Simple as that. So let's actually do the math for alignment 2. We've got 6 spots that match perfectly. At plus 2 points each, that's 12 points. There's one mismatch, so we subtract 1 point. And we had to add 2 gaps, which cost us 2 points each, so that's minus 4. So what's 12 minus 1 minus 4? It's 7. The math tells us, clear as day, that alignment 2 is the better hypothesis. But wait, is a score of 7 good? A high score is great, but it's pretty useless without one more critical piece of information, the E-value. Now, this might be the most important number you see. It answers a simple question. What are the odds that I would get a score this good just by pure random luck? And you want this number to be tiny, like ridiculously, mind-bogglingly small. The gold standard is usually anything less than 10 to the minus 4. That's 0.0001. A number that low is your statistical proof. It's the universe telling you, hey, this is almost definitely not a random fluke. You've probably found a real biological connection here. Okay, so let's zoom out. We've gone from the why to the how and all the details in between. What's the big picture here? What can we actually do with all this? So let's wrap this all up. We started by seeing that alignment is our main tool for digging up evolutionary clues. We hammered home that critical difference. Homology is way more than just similarity. It's a conclusion about a shared past. We saw the choice you have to make. Global alignment for the big picture. Local for finding the hidden gems. And finally, we learned that the magic combo of a high score and a super low E-value is the sign that you found something real. And that's really what this is all about. This isn't just about algorithms and scores. It's about figuring out how to read the library of life itself. We are unlocking the history written inside our own cells. And that just leaves one last incredible question to think about. If our DNA really is a history book, what other amazing stories are just sitting there waiting for us to turn the page?